I'd like to start with a question to you, which is not for marks, but this is something that's always fascinated me, and it's something that lately gets my hackles up quite a lot. If I can just ask briefly, by a show of hands, who thinks that kwashiorkor is caused by low protein intake? Nobody, okay. I think that's the end of my lecture then. Clearly, I don't need to be here. I get lots of people telling me that it's caused by low protein intake. Some think it's aflatoxins. Anybody for aflatoxins? Nobody? Poor hygiene, recurrent gastroenteritis, diarrhea? Yeah, maybe. Okay. Insufficient antioxidants. I'm sure there'll be some hands for that one. No, nobody. Okay. What about a mismatch between the intrauterine and extrauterine environments, pretty much like the Barker hypothesis? Yes, Anna-Marie goes for that one, thank you. Who thinks it's detrimental bowel flora? No one. Okay, so there's a lot of people sitting on the fence, clearly. So hopefully there will be some points to my presentation. When I was looking or making this presentation, I found these two quotes. Now, the first one is from Wikipedia, and we all know that we shouldn't trust Wikipedia, so I didn't really look at this, but here it is. Wikipedia says a lack of protein in a South African's diet can lead to forms of severe malnutrition known as kwashiorkor. Okay, that was on the 13th of August. So, but far more reputable is up-to-date, and I think a lot of people use up-to-date because they've got excellent articles on more or less every medical condition you can think of, and they're reviewed annually or sometimes twice a year by the experts in the world. So this is really disappointing. Kwashiorkor is characterized by muscle atrophy with increased body fat or normal body fat. It's caused by inadequate protein intake in the presence of fair to good energy intake. Okay, don't believe it, and let me tell you why. So we're going to start off with a bit of history, and I'd like to just mention Cicely Williams, or Cicely Williams, I'm not sure how you pronounce it, who was one of the first female pediatricians, I think, on earth. She's got a very interesting story, and there's actually a biography out on her, which I haven't read, but I'd really like to get my hands on it. She was born in Jamaica, and then apparently at age 9, her father told her that, look, you're never going to find a husband, you better become a doctor. And so she did. It was actually in the First World War. They were in England by that stage, and all the men were off to war, so they were looking for people to study medicine. And that's how she got into medicine, and she became a doctor and later became a pediatrician. And then when she qualified, the war was over, so all the men came back, and she was not very popular because no one wanted to employ a woman doctor. So she went on and eventually ended up in Ghana. And she coined the term kwashiorkor from the Ga language. And so that story is famous now, but I think at the time she was very much shunned, and people didn't believe her and didn't want to pay too much attention to her because she didn't wear pants. So she was actually the one that started this rumor that it's protein deficiency. She said that it's notable that all these children have a very monotonous diet based on cereals. And from there were some observational studies that showed that indeed these diets didn't have enough protein, because protein, as we know, is the most expensive part of many diets, or in terms of macronutrients. Then later on, people found that hyperalbuminemia was very common in children with kwashiorkor. So it must be true, it must be protein. And as we saw, still a lot of people believe that it's protein deficiency. And I was recently overseas for 2 months in a gastroenterology unit, and similar there, people quote or just say, yes, you guys see kwashiorkor, which is if you don't eat protein. And same on the ward rounds here in South Africa. My registrar tells me the same thing. So we've got a lot of educating to do. So in about 68 was the first question raised about this, when people showed that if you compared the diets of children with quash and marasmus, in fact, their protein content didn't really differ. At the time, also, there were some papers from South Africa supporting this protein theory, because they showed that children with quash-fed amino acids actually lost their edema and improved. And at the same time, their albumin went up, so clearly it must be protein. But then there was this guy called McLaren who published in 1974 in The Lancet, The Great Protein Fiasco. And he basically showed that, in fact, there were no differences in protein intake, and that you didn't need protein to resolve kwashiorkor, etc. And a great debate ensued with lots of answers and letters to the editor in The Lancet. And the question was raised. I think John Waterlaw is another very important name in the research on kwashiorkor and on malnutrition in general. And they showed in Jamaica in '76 that actually there's no evidence that protein deficiency is the cause, and they raised a big question about it. So they also showed that your albumin responds not just to protein intake, but to other foods as well. So the protein theory was out of the window. So same group in Jamaica then went looking for other explanations, and Michael Golden and his colleagues identified this theory of the low antioxidants, and it is an imbalance between free radicals and antioxidants in the patients with kwashiorkor. They found in trials that the children with edema had low glutathione, which is an antioxidant, and other antioxidants, and that the infections and toxins that are frequently encountered in children with kwashiorkor produce this pro-oxidant state, and therefore the excess free radical damaged the capillaries, and they got edema, and they almost look like septic patients. That theory was also shot down, but this is a lot more recent. Gilberto did this big trial in Malawi, and I'm quite proud of finding this little cartoon, because I think this guy looks very Italian. Is that point? No. Okay. Well, anyway, he threw a spanner in the works. He did a large trial. Oh, that's one. Thank you. He did a large trial, 2,300 kids who didn't have kwashiorkor or malnutrition, and he gave them all antioxidants or placebo. And he showed that the incidence of kwashiorkor was actually slightly higher in the supplemented group, although it's not statistically significant. So clearly, antioxidants do not protect you from getting kwashiorkor, and that put an end to that theory as well. So the majority of the time I'm going to talk about what the current theories are and what people believe. And I know that I've skipped out on some other theories like the aflatoxins, but suffice to say that not many people believe it. Okay, I'll give you a moment to read the cartoon on theories. So what is it? We all agree it's insufficient nutrition that causes malnutrition. What else has got to do with it? Is it epigenetics? Is it enteric infections? Or is it, in fact, this microbiome which we're hearing so much about lately? So we're going to start off with epigenetics, and this is a bit of a short section, but there's an interesting paper that came out. I've forgotten when it was now, I think 2012, last year, where people analyzed data from that large Jamaican study group retrospectively. And they took 1,300 cases of severe malnutrition over the 30-year period there. And they looked, and they found that their birth weights differed. So if you look at the marasmic group, they had a lower birth weight than the kwashiorkor group. There's a lot of overlap, but there's a statistically significant difference between the groups, and marasmic quash is sort of in between. So they found this theory that maybe this is the same as the Barker hypothesis. So in the Barker hypothesis, we think that babies who are undernourished in utero, maybe their placenta wasn't working so well, they're born small, then they gain weight rapidly, and later in life they all get diabetes and heart attacks and obesity, etc. This is very beautiful. It's the opposite. Babies that are well fed in utero, okay, the large or the normal gestational weights, then get faced with starvation outside of the womb, and therefore they haven't adapted to this environment, and they have a maladaptive response to starvation, and they develop kwashiorkor rather than the marasmic babies who just shut down their metabolism and use all their nutrients very sparingly. So this is an interesting theory which hasn't been tested further, but certainly fits in nicely with the current theories on the Barker hypothesis. So the second topic is a bit larger. It's enteric infections and intestinal inflammation. So there's a few people which I want to introduce you to who believe that malnutrition is actually an enteric disease, that it's a disease of the bowel primarily, obviously on top of a bit of malnutrition. This slide is something that you can find in many different papers, and all it's showing is that as you have more days of diarrhea or more episodes of diarrhea, you're more likely to be malnourished. And there's many different trials that show this, and I think we're all quite familiar with this. This is Gerent, who's one of the proposers of this, and his paper, Malnutrition as an Enteric Disease, has this picture in it which explains it quite nicely. So we start off with pathogen ingestion and enteric infections, which may or may not cause diarrhea. Okay, so we're not just talking about diarrhea, we're even talking about subclinical infections in the gut. One intervention we could do at that stage is antimicrobials, but in the vast majority, and I'm certainly not saying that children with diarrhea should get antimicrobials, but in these specific infections it might help. So they don't get antibiotics, they get intestinal damage and inflammation, which I'll show you more about. And therefore, they lose nutrients like protein-losing enteropathy, and they also malabsorb other nutrients. If you have enough micronutrients, you can repair that mucosa, like zinc, like other vitamins. You're going to have an improved repair of the mucosa, but if you don't have that, you're going to continue losing nutrients, and you're going to get malnutrition, which we know impairs your immunity, which worsens your infections and puts you at risk for more infections, especially if you live in an environment with unclean water, etc. So that's the whole theory behind why enteric infections can cause malnutrition. So here's one trial which kind of shows a model in a mouse with, where infection can cause malabsorption in the gut. So what we see here is a normally nourished mouse, and they took the small bowel and they've made a nice little histology slide of it, and what you can see is there's very large villi and everything looks nice and happy. If you take a malnourished mouse, you see that the villi are far more disorganized and not as tall as the normal mouse. If you give a normal mouse Giardia, the Scriptosporidium, so a parasite which causes diarrhea, especially in immune-compromised patients, you see that, again, the villi are not very well organized, and they look more or less the same as a malnourished mouse without infection. But if you give that malnourished mouse the Giardia infection, they end up with a lot of villus atrophy and a lot of damage to the bowel. And you can imagine that there's a lot of leakage of nutrients out into the bowel lumen, and that there's poor absorption of nutrients back in. And this is just a picture showing the different response to feeding these mice. The normal control mice feed and gain weight quite rapidly, whereas those that were malnourished and were infected do a lot worse than all the other groups. Okay, so again, it's just establishing a link between infection in the gut and malnutrition. There's also other infections, and the ones that have been blamed most are intra-aggregative E. coli, which is quite common in developing countries, Giardia, which is this lovely parasite there, which we think is quite common in our setting, but we have difficulty proving it because you really need to get the stool samples very quickly to the laboratory to look under a microscope to see this bugger. Then we've got ascaris and other helminthic infections, and they've all been linked to disturbed intestinal function and poor growth. Okay, I've mentioned that you don't necessarily have to have diarrhea to have these effects and malabsorption in your bowel, and that you might not always know that it's there. Then I want to introduce you to environmental enteropathy. And this might not be an introduction. I think a lot of people should have heard of environmental enteropathy, but it's not very well studied. So environmental enteropathy is a spectrum of small bowel diseases, essentially, which lead to inflammation, decreased absorption, increased permeability, and it's very common in people in developing countries. Okay, it's also known as tropical sprue, and it's if you look at it macroscopically, so through an endoscope, it looks very similar to celiac disease. This is tropical enteropathy or environmental enteropathy, and what you're supposed to see is that the villi sort of fuse and form leaf shapes. It might be a bit difficult without a picture of a normal duodenum, but it looks similar to celiac disease, and microscopically as well, actually. So what does it do to the intestinal barrier? And this is a picture just highlighting the different layers of mucosal defense that we've got, starting on the inside with the normal flora, which we know protects us from abnormal flora and pathogens. Then we've got this mucus layer, which is divided into stirred and unstirred, and there's lots of antimicrobials in it and mucins, etc., which prevent these bugs in the lumen from attacking our cells. Then we've got the intestine ourselves. The villus cells have got microvilli, as you know. There's tight junctions between the cells which get broken down in environmental enteropathy. And then below that, we've got the lamina propria, which has got a large immune constituent and lots of immune cells in there preventing infection. So the increased permeability that we see leads to several things. The one way we can test for it is a fractional excretion of lactulose and mannitose. It's not a great test if you're looking at individual patients because there's a lot of variability, but basically lactulose, as you know from treating constipated children, is supposed to not be absorbed in the gut. If you've got increased permeability, it is absorbed, and you get some excretion in the urine, and that's how we know that there's increased permeability in this gut. The problem is that this leaky gut then predisposes you to translocation of bacteria and endotoxins, and when you test the blood of these children with these leaky guts, you find a lot of lipopolysaccharides which belong to the bacteria actually in the blood. The environmental enteropathy appears to be seasonal, and it's linked to the pathogens that we've mentioned, and it leads to chronic inflammation, and it is a feature of HIV infection. So these are some of the things that we think predispose you or cause the enteropathy: HIV, abnormal microflora, poor sanitation, diarrheal pathogens, and a genetic predisposition lead to the inflammation, decreased barrier function, increased microbial translocation, and therefore we can imagine that the malnutrition that goes along with it can also present as kwashiorkor with the translocation of the bacteria and the bacterial infections which we frequently see in kwashiorkor. Okay, this is a recent paper from quite an interesting group, and they've got a website if you just Google MELED, which is a group of countries across the world that are doing research into this environmental enteropathy. This particular paper, they analyzed stool samples from children at different ages, and they measured some markers that marked protein-losing enteropathy and inflammation, basically. And those with more inflammation grew less over the next 6 months. So again, it's linking this inflammation and this environmental enteropathy to poor growth. What about treatment? That's the sad part. We don't have any trials that have shown to be effective, but there's lots of trials happening. That's the website, by the way. Rifaximin is an antibiotic. They did an RCT on this, a randomized controlled trial, to see if we give this, will it prevent environmental enteropathy and will it improve nutrition? Unfortunately, it doesn't. The ProNut study was a study of pre- and probiotics in severe acute malnutrition. And unfortunately, those kids didn't do better than if, whether they got prebiotics and probiotics or not. And there's other trials going on, all trying to interrupt this vicious circle of environmental enteropathy and malnutrition. Okay, the hot topic at the moment is the microbiome. And there's a lot of research going into this and how the bacteria in our gut interact with all sorts of diseases and conditions in the human body. So I don't think I need to tell you this, but there's more cells in our gut, bacterial cells, virals, and helminths, than there actually are in the entire human body. There's a huge number of species in there. And what we know from our undergrad teaching, I'm sure, is that they are responsible for fermenting some unfermentable polysaccharides. Those form the short-chain fatty acids which feed your colonocytes. They're also known to deconjugate bile salts, and they synthesize some of essential amino acids and vitamin K. We were never able to study these bugs because they're very difficult to grow in lab conditions, and there's so many of them, you can appreciate that it's almost impossible to start that task. So, but since new techniques of DNA sequencing, we've been able to identify a lot more species. And the metagenome, which is the genome of our microbiota, is becoming more or better understood. And this has given us new insights into the pathophysiology of different diseases. And obviously, malnutrition comes into that, but there's many other diseases that we didn't previously understand. They weren't explained by genetics alone. They weren't explained by nutrition alone, and now we can link the two and we can find out more about them. So the studies usually rely on twin pairs, because twin pairs you control for the genetic material. Identical twins have got the same genetic material, so they give you a great opportunity to study. And then this thing called gnotobiotics, which are basically animals grown in a sterile environment. So usually mice, they're born and they're never exposed to any bacteria or virus or anything at all, and you're then able to interfere with their microbiota as you please. So those, are the two concepts that we need to just keep in mind. And the example I'm going to briefly mention, this was a big paper in 2006 in Nature, which showed that obese mice and humans have different microbiota compared to their lean counterparts. And both in the humans and the mice, there was a difference in the balance between Bacteroidetes and Firmicutes. And this was seen in both groups. Then they took, they looked at how these mice became more obese, and it seemed that they're better at extracting energy from food if they've got this microbiota in their gut. They then took the poor mice that haven't been colonized with any bacteria, and in this case they took leptin-deficient mice. So these are mice that are prone to get obesity because of a deficiency of the leptin hormone, and half of them got fecal transplantation from obese mice, and the other half got fecal transplantation from lean mice. And those that got the obese microbiota got a lot more obese than the ones that got the lean microbiota. So this is proving that, in fact, the microbiota itself is certainly part of becoming obese in these animals. So obesity became an infectious disease. Soon thereafter, there's this very elegant trial from Smith and colleagues. And he followed 317 pairs of twins from birth until they were 3 years old. Some of the twins were monozygotic, identical. Okay, we said that's important because they've at least got the same genetic material. And out of those 317 pairs of twins, 50% remained well-nourished, but 50% got severe malnutrition. Okay, so that's quite an indictment for the poor Malawian twins who seem to be very predisposed to becoming severely malnourished. And what they saw is that the majority of these, only one of the twins became severely malnourished, but in 7%, both of them became malnourished. So they treated all the malnourished twins with ready-to-use food, and if they were discordant, i.e. one twin was normally nourished, then both of them got ready-to-use food, because otherwise they probably would end up sharing anyway. And then they studied the metagenomics of these children. This is a picture just showing you overall with age that this marker, PCI 17%, I don't know what it means, but basically it's a marker of maturity of the microbiota. And you see, as the children get older, the microbiota seems to mature. This is a picture of two healthy twins that did not get malnourished, and you can see that with time, their microbiota matured. But this is projects really badly, sorry. This is a picture of a healthy co-twin. Again, you see that maturation of the microbiota. And this is a picture of a twin that got kwashiorkor or severe malnutrition. Yeah, it's kwashiorkor in this case. So you can see that there was no maturation of this microbiota. So in the first part of this trial then, they showed that there was something in the microbiota that was different in the twins that got malnutrition compared to the ones that didn't get malnutrition. They then took it further exactly like they did in the study with obesity, and they took fecal samples from 3 of the twin pairs that became discordant. Okay? So you've got one twin that's healthy, one twin that became malnourished. And they put it into gnotobiotic mice again. They fed these mice Malawian diets, gave them ready-to-use therapeutic food, and then normal Malawian diets again. And again, they followed the metagenome. And this is a graphic presentation which always shows it best. This is a mouse before the experiment, gnotobiotic, so it doesn't have microbiota, and they gave it a fecal transplant of a healthy child. This mouse got the fecal transplant of a child with kwashiorkor. Then they both got Malawian diet, and the one that got the kwashiorkor microbiota became malnourished, and the other one didn't. They gave ready-to-use therapeutic food, and the mice grew. But what's quite worrying is once they stopped the ready-to-use therapeutic food, which is how it happens in most situations in countries where they treat malnutrition with ready-to-use food, that mouse once again became malnourished. I'll skip that for time's sake. So very interesting. Kwashiorkor and severe malnutrition also seems to be, at least partially, an infectious disease with regards to a microbiota. They also found other things in the mice with the kwashi microbiota, got some products that interfere with energy metabolism. And what we've always known is that there's interference or malfunction of energy metabolism in cells in people with kwashiorkor, which causes them to get leaky capillaries and causes them to have edema. So this might be one mechanism how these children with kwashiorkor actually get that abnormal energy metabolism in the cells. Okay, we've mentioned that ready-to-use therapeutic foods had a transient response. Okay, this is the last thing I want to mention. Another article that's maybe interpreted in different ways, I think. And it was a trial that looked at antibiotics in children with severe malnutrition. Big trial, good quality, double-blind, placebo-controlled, 2,700 kids, and they treated all of them with ready-to-use therapeutic foods, and then one group got a cephalosporin antibiotic, one group got amoxil, and one group got placebo. And what we see then is these really predict badly, I'm sorry. The children receiving, or the overall mortality didn't really differ between the groups. But if you look at the recovery rate, it was far quicker in the group that got cephalosporins or amoxil than in the placebo group. So you can explain this one of two ways. This could be because most children with malnutrition are at high risk for other infections, and therefore we're treating them, and therefore they're getting better quicker. But the other interpretation would be that, in fact, you've interrupted the cycle of environmental enteropathy, or you have treated the overgrowth of microbiota that's not beneficial to the host, and therefore they recover quicker. So quite an interesting different or what you take out of the different stories. So that's what I had to say. I think it's quite amazing that a disease so old, we still don't really understand the ins and outs of severe malnutrition and what causes it. But this opportunity really opens up things for the treatment and the prevention of malnutrition in the future. And the more we understand about this clearly, the better we can prevent this disease, which still carries such high mortality and is still so common in sub-Saharan Africa. So who knows, maybe in future we'll be treating kwashiorkor with fecal transplants. We'll see. Thank you.